Yes.
Wait. Okay. Okay. Well, good morning, everyone. Those who are in the US, and I have to start by saying that I'm very happy to be part of this conversation. I'm a kind of newcomer to have this very general and global, say, ways of addressing problems. And well, I'm going to make my best to fit our own work and our own interest in the context of what we have been discussing these days. Okay, so let me just tell you that I come from Madrid, from the National Center of Biotechnology, and I just want to show you the size and the faces of people working in my group. Okay, so I'm going to discuss with you today ways of tackling pollution. And obviously pollution might be something that is a multi-scale phenomenon. So you can go from very kind of located site all the way to a global dimension. But, you know, whether you go in a lower scale or a bigger scale, you always face the issue that has been repeatedly addressed in this meeting about tipping points. So that means that if you have let me see if I get the laser pointer. Yes. So you have a situation of stability and you stress the system where you have a biological, say, balance. And then you are within some limits, then you can argue that if you stop the stress, then the system will return to some degree of stability. But if you overdo and you go beyond the tipping point, then you go into another regime, or you can screw up entirely the whole thing. And then what happens is that you have to apply a more intense and elaborated medicine. Like when you have a very mild disease, you can take aspirin or you can take ibuprofen, but you have a very serious disease, then you have to kind of think on very sophisticated ways of returning to a healthy situation. So the starting point of all this story I'm going to tell you today is that, well, when you have a kind of a big problem, in my opinion, the way to address it is to divide it in a smaller number of tractable problems, because otherwise, in many cases, you don't have a global solution for everything at the same time. So what are these list of problems that are connected to global, say, environmental deterioration? Well, you have a list of them there. So obviously, one of them, and maybe the most important, is the growing atmospheric levels of CO2 and greenhouse gases. But you have many others, and some of them have been tackled in the conversation these last few days. So we have the expansion of drylands and soil desertification, one of the favorite topics of Fernando. Then we have plastic waste in marine ecosystems. We have micropollutants in trophic chains. We have the problem of nitrogen and phosphorus. We have the problem of air pollution. We have the problem of excess of lignocellulosic waste, you name it. And then you have problems with scorched soil and so on. Okay, so what we have been doing so far from the scientific and biotechnological point of view. Well, what we are doing and have been doing for a long time is to remain in this kind of scenario A, that is before the tipping point. So, and to do that, or the way that people are happy under such a scenario, is to try to reduce CO2 emissions and go into an energy that has less carbon, or to produce biofuels, or to improve crops and all that. But these are all what you may call reactive approaches. So the question is, can we be proactive? I mean, can we really do something to address cases where we go beyond the tipping point? Okay, years ago, and under the kind of mastermind of Ricard Solé, then we and others started to discuss the possibility of, thinking big. And instead of thinking in bioremediation interventions, just by focusing in a small place with a petroleum spill or some metal spill or something, try to think whether you can develop technologies, actual technologies, not theories, but actual technologies that allow us and enable us to address these big, massive environmental pollution problems. And this is what I'm going to discuss with you today. Well, the bottom line is something that was mentioned by Ricard in his introduction, and is the idea that to make a difference and to have an impact in these situations, we need to design agents, and agents that get implicated into the ecology of the sites and have the capacity to change the entire equilibrium by entering into the system activities that are missing or have disappeared because of the human action. So, well, there are a large number of naturally occurring ecosystem engineers, and we have been discussing in part some of them in past few days. But the kind of a challenge and opportunity of synthetic biology is to see whether you can go to the laboratory and rationally design biological agents that you engineer specifically to deliver the activities that are necessary for recovering an otherwise deteriorated ecosystem. And to do that, and this is a picture that comes from a recent paper with Ricard and his collaborators, well, you have to start thinking on some kind of block diagrams to figure out what type of relations are necessary to have a stable and well-balanced ecosystem. Then you think or try to survey what biological, say, parts or what biological components or actors you need to implement these blocks in real world. And then you can abstract these activities in the shape of logic interactions, and eventually everything boils down to writing a DNA sequence. Well, this would be like the dream or the ambition of synthetic biology. And this is exactly the type of kind of proposition that from this community of synthetic biologists we offer to people like you and people who are interested in more global environmental scenarios. But the DNA has to go somewhere to be effective and to release all the information and to actually implement the activities that we are after. So what choices we have to put the DNA in for developing these ecosystem, catalysts? Well, some calculations have been made on what are the main actors, biological actors that we find in the biosphere, and who are the actors that produce more biomass or less biomass. And then, well, some calculations that were made some time ago by Ron Milo then indicated that the bulk of the biosphere biomass is produced by plants. But there's some qualification that we have to make to this statement, because you have to be aware that about half or more of the entire biomass of plants is just one molecule, that is cellulose or lignocellulose, and therefore this is a molecule that doesn't have any interest from a catalytic point of view. And therefore, if you look at the map of different biomass actors in the biosphere, you end up realizing that bacteria are the ones that really can be useful for this type of purpose. And this is because of two reasons. One of them is because there's a lot of bacterial biomass in the oceans, in soil, all over the place. And in terms of the substrates and volumes and weights of products that can be handled by bacteria as a global catalyst, you realize that only the environmental microbiome can make it. So you make some calculations, and this is a calculation that I made some time ago. Then you can go from the same bacterium all the way from a test tube all the way to a very global, say, activity. And you notice that, in fact, in terms of the activity and in terms of the biomass and quality and quantity, the environmental microbiome is our only potential ally to really deliver activities that could be useful for recovery of ecosystems. The other thing, and this is something that is very dear to my heart, and also I'll try to convince you that is excellent as a kind of technological asset, is that DNA propagates very, very quickly. So we know that because of the spread of antibiotic resistances and the spread of pathogens. But a horizontal gene transfer doesn't have to be related necessarily to bad things. So we think on, let's stop horizontal gene transfer because it's very bad for antibiotic resistances, or because you spread pathogens or whatever. Well, this is true, but at the same time, the same mechanisms can be recruited and can be domesticated with the tools of synthetic biology precisely for a good purpose, namely for spreading beneficial traits in a larger microbiome. So think on this as a kind of vaccine, So if we kind of vaccinate the environmental microbiome for degrading this compound, or for fixing more CO2, or for retaining more phosphorus, or other activities of environmental interest, then you may end up with a situation in which the entire, say, biosphere and the entire microbiome benefits from this massive horizontal gene transfer. So don't think on horizontal gene transfer only in terms of bad occurrences. Okay, so you can make then a list of technical challenges that you have to overcome for this type of applications. And then, well, you can divide them in various categories. You have scientific questions, for instance, how to optimize pathways. And this is something that keeps, I would say, 80% of synthetic biologists busy. you engineer a pathway for degradation of this or for production of that. How can you optimize these pathways for production or degradation of something? This is a big part of contemporary synthetic biology, but there are many other kind of lateral aspects like interoperability of the pathways, expression systems, stability, and obviously we need also ecological and theory models. And then we have enabling technologies, and the say star enabling technology these days is obviously DNA synthesis. As you know, now we can synthesize genomes, entire genomes, and we can program entirely the genetic material of a given microorganism. And this is something that in the future will become easier and easier. But there's this other aspect that is not really developed, in my opinion, however, is the key for having this possibility of delivering activities at a very high scale. And it's what I like to call environmental galenics. So what do I mean by environmental galenics? Well, environmental galenics is about the following. You may go to the laboratory and produce in the laboratory a wonderful catalyst that fixes CO2 or inactivates this pollutant or degrades plastics or you name it. However, one thing is to have an activity working in the laboratory, and a completely different problem is to have it working at a very, very large scale. So this is where environmental galenics comes in. So galenics is this idea or this concept that comes from pharmacy, and is this challenge of how to deliver an active principle to a sick body. So as you know, in medicine, you can deliver the same active compound through many different means. I mean, a given antibiotic or a given molecule can be given through a pill or an injection or an aerosol or a suppository. And at the same, but it's the same thing, but it's just that you have to think on the physical format, the material format that you need to engineer to have this introduced into the sick body for this molecule to have an effect. So by the same token, we argue that we need to think not only on the activities that would be necessary for helping the environment in various aspects, but also how to deliver them into this environment and what is the format, what technology we can think of to really spread these activities at a very, very large scale. Well, the question number one is, what do we want to deliver into the environment? And well, and if you think on bacteria, you can think in various possibilities. You can think on monoclonal agents, and it's one strain, for instance. In many cases, and I will try to argue that later, it's better to think on meta-organisms or design microbiomes. So this is because natural evolution has told us that when a community or a bacterial, say, agent faces a new challenge, normally it's not that the job of one specific strain to deal with the challenge is generally the result of the division of labor in a wider and more diverse community of actors. And this is something that perhaps we can bring into our design or synthetic biology design agenda, and thinking on agents that are not monoclonal but polyclonal. But also, and we'll return to that later, we can think on spreading genes. Instead of thinking of inoculating different ecosystems with one strain or with a consortium, you can even think on just enriching and fortifying a given environmental microbiome with a new activity just by introducing massively new genes into that particular consortium. I will discuss all these things little by little. Okay, well, number one, this is a starting point. You have to, we have to think on, as a starting point, identify a good chassis. And chassis is a magic word in the jargon of synthetic biology, and is inspired in this idea that you can have a car or some vehicle and you have like some basic functions that are in many cases not seen, but then you have like a framework on which you can put things in and out, and at the end have a device or a system that will respond to your specifications. So by the same token, well, you can think on what type of bacteria could be interesting to start as a chassis for this type of environmental applications. Well, you look in the literature, in the synthetic bio literature, there is a whole bunch, a whole collection of environmental bacteria that have been proposed as chassis for this type of applications. And I will just mention two of them. One of them is or are cyanobacteria, and people in this audience, some of you have some experience with them. Cyanobacteria can be wonderful agents for environmental remediation and environmental applications. Their little problem is that their genetics and the tools for their genetic programming is a bit complicated because they have some issues that I would not discuss now. but still it's a potential and very useful chassis to think on the future and even today for this type of applications. And also Fernando and Ricard have been playing with this idea for some time. There's this other type of bacterium that I like very much, and that are Pseudomonas. Pseudomonas is a very wide and broad, say, type of bacteria. And there's a branch of them, the ones that are called Pseudomonas putida, that happen to thrive and to live happily in soil polluted with contaminants. And this is something that for us is very attractive, of course, because in principle, you think that this chassis is already habituated to live and to exert and execute its natural activities or enzymatic catalysis or whatever in the very same places where we want to kind of remediate a given environmental or pollution problem. So you can start the process of converting a natural isolate into a chassis by just looking for this type of bacteria in the location that is afflicted by the environmental problem. And then you take it to the laboratory, you sequence the genome, and then you identify what things are interesting to keep and what things are interesting just to get rid of. And for instance, in the case of Pseudomonas putida, there is a soil bacterium that is habituated to overcome stress and has a very robust, say, lifestyle. There are some good traits. For instance, is tolerant to solvents. It has a metabolism without of any DPH. It's a safe host for recombinant DNA, and also it has some bad traits. It's resistant to some antibiotics. It has some metabolic problems. It needs oxygen for growth. And also, in this case, is a close relative of a pathogen, that is Pseudomonas aeruginosa, that I'm sure all of you have gotten at some point in your life when you got an ear infection or something like that. But still, this branch of Pseudomonas are nice guys that we can use and recruit and domesticate for the type of applications that I'm talking about. So what is involved in domesticating or starting with something that is wild, and how can we really convert that into a chassis for these niche creation applications that we are after? Okay, then the challenge can be named under the paragraph or under the title of engineering, biophysical engineering of synthetic biology agents. So what would be an ideal synthetic biology agent? What type of formulation would you like to have in one of these agents? Well, you can argue and you can propose various scenarios, but one that is very attractive is the one that you see in the screen. Namely, that you have a consortium of bacteria A, B, C, D, when you have a division of labor, so for a given purpose. So imagine that you want to degrade a given compound, say maybe bacteria A and B gets rid of the compound, bacteria C eliminates a toxic intermediate, and bacteria D eliminates a possible predator. So, and you can always think on developing consortia that are specialized in covering various challenges that bacteria find where they are in the environment. And at the same time, you may want to have them in a particular, say, three-dimensional structure in a space. Okay, so and to do that, you have to think on how to, make sure that they touch each other with a particular, say, a code of contacts. So at the end, you have a given three-dimensional structure. But then at the same time, you may want to have this consortium sticking to a surface. And if the surface, for whatever reason, has also some catalytic efficiency or some catalytic activity, so much the better. However, when you face environmental bacteria, what you face is this very, very messy situation. Environmental bacteria are not domesticated and they stick to whatever they want and they have their own biological agenda. So what can we do to domesticate bacteria, at least, to start the process to convert them into a good chassis and to, be amenable to some engineering, not only from the biochemical point of view, but also from this physical point of view. Okay, so the ultimate objective would be to have tools to come up with these three types of scenarios. For instance, you may have a monoclonal catalyst here, where you have the bacterium engineer for degrading something, or you have a synthetic consortia between bacteria, with bacteria and fungi, or bacteria and yeast, you name it. Or at the end, you may want to have also the possibility to stick these communities to a layer or to a solid surface. And then you have a different problem, and it has to stick bacteria to a, say, solid, say, target. Okay, so what can you do? Well, and now what you do is that first you see what is the starting situation, and the starting situation is that bacteria have their own attachment preferences. They have a bunch of different structures on the surface that allow them to stick to their favorite places. And you have all these protuberances that obviously will not be very helpful when you try to develop nice three-dimensional structures of consortia. So one possibility, and this is something that we have explored in our laboratory, is to produce what we call naked bacteria. So naked bacteria are bacteria in which we edit the genome to delete or to eliminate surface structures that interfere with our own programming purposes. And then at the end, you find a cell that has a different, characteristics and different qualities on the surface as compared to what you had before. So what you do is that it's like, you take the bacterium to a hairdresser, and then, you start cutting the hair, and you have to put some kind of fashionable surface and so. And at the end, what you have is a situation in which you go to the genome and eliminate all the genes that encode all these surface structures, just to have at the end something that is more amenable to engineering. This is one of the examples of naked bacteria. So in this case, this is Acidomonas putida, in which you have eliminated all these surface structures. And unlike this wild type situation where you have your surface packed with villi and hair and flagella and you name it, in this case, you have a very smooth surface that can be used as the scaffold to program the cell to do other things. Because the advantage of undressing a bacterium is that you can redress the bacterium with other things. And this is wonderful because it's like you have a little puppet, and you have like the body of the puppet, and you can put different kinds of decoration in the puppet. And obviously all this can be made genetically, all this can be programmed genetically, and all the instructions of all this dressing be put in the chromosome, for instance, and or synthesize the DNA or to write the sequence or whatever. So one example, just one of them, we have many of them. We have redressed the surface of Pipitida with a cellulosome or with components of a cellulosome. So cellulosomes are very complex protein structures that are found in naturally occurring cellulose-degrading bacteria. And they are complex, and they follow a very, very interesting, say, Lego type of structure with standards where you can put things out and develop and have very, very efficient external cellulose degrading machines at the end of the day. So what someone in my laboratory made, for instance, was to engineer the surface of Pseudomonas putida, of this naked Pseudomonas putida, and redress it by genomic encoding of the properties with a multi-protein complex that has some activity on some cellulose components. And this is something that is very nice, because if now you engineer in the cell the possibility to degrade, for instance, glucose and xylose and arabinose, and so then you have a super cellulose degrader, and this is a combination that nature has not really invented very efficient. But you can redress the surface with other very interesting things. For instance, you can redress them with activities to capture residual water. And this is something that, for the sake of bringing some little humidity to arid lands, can be very, very interesting. And one of the projects that we have in the lab, for instance, is to see whether we can recruit the structures that are found sometimes on the surface of these desert lizards and the spikes and so, that have a tremendous ability to capture residual water, and see if we can transplant them and put them on the surface of some soil bacteria, for instance, Pseudomonas, and see whether by doing that you can increase marginally the humidity of soil, and maybe by doing that you start generating a new niche that helps to reconstruct an ecologically balanced, say, system. Okay, so but this is what you can do with standard monoclonal bacteria, but as we mentioned before, sometimes the idea is not so much to have monoclonal, but to re-engineer a whole microbiome. And well, this picture is taken again from one paper from Ricard, and where he and his colleagues provided some, say, background on the basis that having engineered microbiomes can have a superior efficiency in terms of reconstructing a deteriorated ecosystem as compared to just having a monoclonal strain. If in the system you introduce cyanobacteria, so much the better, because the cyanobacteria will capture light and will start kind of delivering nutrients and delivering other, say, carbon, say, resources that will be useful for maintaining the activity of the other members of the community. And so this part of this challenge of microbiome engineering is a very, very interesting, area of synthetic biology right now, and a bunch of laboratories are trying to develop tools and concepts to really engineer good microbiomes, in principle for medicine, but also for environmental applications. This is a paper that was shown also by Ricard, where you can see that people working, for instance, in coral bleaching, entertain the possibility of going to the laboratory, develop synthetic microbiomes, put them back into the corals, and see whether that can have a beneficial effect for their recovery. Okay, but how can you really put together bacteria that otherwise might not be naturally sticking to each other? Well, one possibility is to, and this is another branch of research in synthetic biology, is to see whether you can decorate a bacteria with artificial adhesins. And well, there are many to choose from. And again, you can encode these adhesins in the genome, and then you can combine the genetic determination of the location of the adhesins on the surface and make some biophysics and some modeling and see what happens when these adhesins are located in the poles of the cell or in the equator of the cells. And then you can end up with the models that will tell you or will predict what type of shapes you can have in a design microbial community. So here you have the possibility not only to combine activities, but also to combine them in a given spatial structure that can be beneficial and synergistic for production of a given activity. So to do that, or a kind of side aspect of that, is that now with this naked strain, you can also make bacteria to stick to surfaces, because if the adhesin to the surface is such that it recognizes something that is in the solid substratum, then you can have a monolayer of bacteria of this type. So one of the adhesins that have been worked much are Camel antibodies. Camel antibodies are naturally single chain. We can go to their genes, put them in the bacteria, and then deliver or express them on the surface of the bacteria and make the whole bacterium to stick to a surface that is coated with the target antigen. So this is something that gives us a tremendous flexibility in terms of what type of target you want to adhere bacteria to. Well, we and others have a pipeline for production of nanobodies, so the technology is already there. And then, I'll show you just one experiment in which, for instance, we try to stick Pseudomonas putida to a surface that is coated with fibrinogen. But it's a kind of model system, but you can use it in any other target. And then you make the genetic construct, and that's kind of cutting and pasting DNA. And at the end, what you have is a bacterium that sticks specifically to a surface that is coated with an antigen and not to any other thing. So that means that technically it is possible not only to engineer microbiomes, but also to put the microbiomes sticking to a target surface. And that's something that for the type of applications that we are discussing can be very, very useful. So let me just finish my little presentation, discussing with you what you may consider like more kind of out of the box possibility, and is this idea of engineering horizontal gene transfer. In other words, that perhaps the ultimate environmental intervention is not so much to put a bacteria into the environment, but to propagate DNA through the environmental microbiome. So the idea in this case is to, to develop in the laboratory a super spreader of DNA and load that super promiscuous DNA with a beneficial activity, and then by conjugation and other mechanisms of horizontal gene transfer, let that to spread through a given population. So this is the idea. So can we engineer a horizontal gene transfer chain reaction? Keep in mind that, as I mentioned before, that we think on spreading good things. I mean, you can think, well, you can spread also bad things. Well, that's true, and this is what nature does already. But we have horizontal gene transfer will happen, we like it or not. So why don't we capitalize on the mechanisms that are out there for horizontal gene transfer and use them as a carrier for kind of spreading beneficial activities? So we have made some progress in that direction, and have put our attention in some plasmids that are out there that are very promiscuous, and we have recruited from these plasmids the promiscuity determinants and reshape them in the shape of a transposon that can jump in many locations. And then when you do that, we have already good evidence that you can really mobilize DNA from different types of bacteria from one place to the other. So this is something that, well, if you are interested, I can give you more details, but this is working very well. And we think that you can really develop systems of massive horizontal DNA transfer and horizontal DNA transfer, chain reactions. Okay, people and ladies and gentlemen, thank you for your attention. Obviously, this is not a finished story. This is something very incipient, and I have to say that whereas the most of synthetic biologists are interested to develop bacteria and microbiomes and synthetic things for the sake of human health or animal health, I think there is a tremendous opportunity to interface with people like you to really develop these ideas where you combine major ecological and global problems with technical solutions that comes from the field of synthetic biology. Thank you very much. That was what I wanted to share with you today.
Okay,